In this screencast, we're going to be talking about enzymes. We know that enzymes are proteins, and these are proteins that specifically catalyze chemical reactions in organisms. A quick review from 2.1, we've got metabolism. So we learned that metabolism is all of the enzyme catalyzed chemical reactions that are happening in an organism. So if it was this cheetah here, we'd be talking about things like cellular respiration or looking at DNA replication or possibly digestion of the meal he's hopefully about to catch because he looks hungry. All right. And we classify our metabolic reactions into both anabolic and catabolic. So anabolic is building. So if this cheetah is building, DNA molecules or building a protein, those are anabolic reactions, right? It's building something. Whereas a catabolic reaction is breaking something down. Maybe it's being digested. Maybe it's being broken apart to make ATP energy, like cellular respiration. So anabolic would be something like photosynthesis, building of a larger molecule, where catabolic is breaking things down into smaller bits. But both of these require enzymes. All of these chemical reactions I've been mentioning require enzymes to happen. Some of them might happen anyway, eventually without the enzyme, but at such a glacial rate that it wouldn't make an impact in the organism overall. So the enzymes allow these reactions to happen in a speedy form, so they're able to access all the molecules and that they need, and to be able to break things down and build things up. All right, so enzymes, as I said, they're catalysts, and they're actually effective in very small amounts. Okay, so you can make just a few of these enzymes and they can do a whole lot. They're unchanged after the reaction. So like any other catalyst, it's not actually part of the substrate or the product. So the substrate is the same thing as the reactant, but we call the reactant in a chemical reaction in living organisms, we tend to call it the substrate. So there's a substrate that's going to be changed into the product. And the enzyme is neither the substrate or the product. It's just helping change the substrate into the product. So it can be used over and over again. And so it's essentially unchanged after the reaction. Okay, so here is an example of a generic metabolic pathway. So if we have starting molecule A, and eventually it's going to be transformed into B and then C, and then the final product is D. So this is maybe building some amino acids that are important to the human body. So it's probably not going to happen just from A to D. There's probably all these chemical reactions in between. Most metabolic pathways are a series of reactions, right? But each one of these steps requires a different enzyme, because we'll see in a little bit that enzymes are very specific to the specific substrate that they're transforming. All right, so this is how enzymes work. They've got this cool structure. It's a three-dimensional structure. They're globular proteins. So each one of these little blue balls in this picture is essentially an amino acid. And we know that amino acid, because they're proteins, a long polypeptide chain or multiple polypeptide chains will be folded up into a particular shape based on their amino acid sequence. So that amino acid sequence will ultimately determine the overall shape of this enzyme. And all enzymes are globular proteins, and they have what's called this specific shape for a specific enzyme. And they have this thing called an active site, and the active site is where the substrate is going to bind within that enzyme. So here's the substrate coming in, and when the substrate gets there, the enzyme is actually going to slightly change shape as the bonds occur between the substrates and the amino acids that line the active site. And that causes a slight change in conformation, meaning a change in shape, and it creates what we call an enzyme-substrate complex. Now that enzyme is going to catalyze a reaction that's going to change the substrate into the product. We'll use some examples like the enzyme sucrase. Sucrase breaks down sucrose into glucose and fructose, because we know that sucrose is made up of glucose and fructose. And if your body wants to break down that disaccharide into the two monosaccharides, here's the sucrose. It'll come in, bind to the active site, and the chemicals, the molecules involved in the sucrose have the perfect complement to the amino acids that line that active site, and then it slightly changes shapes and forms this enzyme substrate complex. And that enzyme substrate complex will allow the water molecule to come in, to allow for hydrolysis. It puts things in the right direction, stresses the bonds just the way they need to be, so it can then break that bond, that glycosidic bond between the glucose and the fructose, to allow those to be individual molecules. So here's kind of the sum of what the enzymes are doing. Okay, so we know it's going to transform the substrate into the product. The substrate binds to the enzyme's active site, and that's going to cause that, form that enzyme substrate complex, right? And what's happening when it's in this, when it's bonded to the active site, is that it's going to orient those substrates in a way, like if it wants to build something, let's say it's an anabolic reaction, it's going to make sure that the parts of those two substrates are put together so that bond can be catalyzed. Maybe it's a condensation reaction. So it's going to orient the substrates the way they need to be. If it's going to break something apart, it could be straining bonds within the substrate. So if it wants to break that sucrose apart into glucose and fructose, it can put some pressure on there. It's a favorable macro environment that can make sure that all of the pieces are necessary for the metabolic reaction to occur. And it's usually covalently bonding to the substrate at that point, so it can really move things around. Once it's done, the enzyme will release the product, and then it can actually be used again. It goes back to its original shape, and then another substrate can come in. So let's take a look at a picture of an enzyme catalyzed reaction. Okay, so here's the substrates in this case. And so the substrates will come in and they will bond in the active site of the enzyme, okay? And it's going to be held into this active site by often hydrogen bonds and ionic bonds, but it can be covalent bonds too. They're held in here. The active site lowers the activation energy. We'll look at that HL in a bit. And then the substrates are converted into the products. So all of that stuff we were talking about, putting pressure on the substrates, orienting them the way they need to be, catalyzing that reaction, then we get the substrates, it releases the substrates, and then now that enzyme is available again, and it can take on more substrates, so it can keep happening over and over again. Enzymes are very specific to the substrate. So sucrase will break down sucrose into glucose and fructose, but it's not going to break down maltose. You need the enzyme maltase to do that. So enzymes are very specific to the particular substrate that they have. Now, there's two ideas about how enzymes work. There's the lock and key model, that the lock and key hypothesis was kind of the original hypothesis about how enzymes worked, and that's that the shape of the substrate was a perfect shape for the active site, and they're complementary to each other. So once the substrate's in there, it kind of clicks in like a lock and key, and then the enzyme can transform it into the products. Sorry. There's also a little bit more adjusted now is the induced fit model, and this is that the substrate and the enzyme, you can see that this has a slightly different shape, and it's suggesting that what happens is that the substrate, when it binds into the active site, that the enzyme actually changes shape to form that complementary shape with the substrate to get the bonds and everything into the right place, to put the stresses where they need to be. So the shape of the active site actually becomes complementary after it binds and can, form that complex, and then it's going to convert it into the products. So there's both the lock and key, and more often now we think about it is that induced fit model where the enzyme active site will actually change shape, this shape change once the substrate is binding with it. Just here's an example of a catabolic reaction, breaking down that sucrose into glucose and fructose, right? And we know that we need water to do that because it's hydrolysis to break it down. And so if this was the sucrose, that would come in, and there would also be water available here. That would bind, make that enzyme substrate complex, and then it will release, get all the bonds oriented where the hydroxyl and the oxygen, can be put in the right spots, and it breaks it apart into the glucose and fructose, which would then leave. Then the enzyme would return to its original shape and could keep on going.